Welcome to GCSE Biology. The topic is about the cell. We will cover this topic from AQA, Edexcel, and Cambridge. If you want to learn about the cell from the beginning, you can watch the previous lecture. Let's continue now. The topic now is scale and size of cells. Cells are far too small to see with the naked eye. We need to use very small units of measurement. The most common unit for cells is micrometer, and its written is meter. A human cheek cell is about 50 to 80 micrometers. A bacterial cell is much smaller, only about 1 to 10 micrometers. Organelles inside cells are even smaller. For example, a ribosome is only 20 to 25 nanometers. These sizes come up in AQA exams, as it is worth memorizing the key ones. Keep in mind that 1 millimeter equals to 1000 micrometers and 1 micrometer equals to 1000 nanometers. Let's now learn the metric prefixes that you must know. Centi means 100. So, a centimeter is 0.01 meters. Milli means 1000. So, a millimeter is 0.001 meters. Micro, written as a symbol, mean 1 millionth. This is the unit we use for cells. 1 micrometer is 0.00001 meters. Nano means 1 billionth. This is used for very small things like molecules and ribosomes. Now for conversion, if you want to go from millimeters to micrometers, you multiply by 1000. For example, 0.08 millimeter multiplied by 1000 equals to 80 micrometers. Going the way from micrometers to millimeters, you divide by 1000. The same rule applies between micrometers and nanometers. Multiply to go smaller and divide to go bigger. Now standard form and order of magnitude. In this topic, you also need to use standard form. Standard form is just a way of writing very small or very large numbers more easily. This format is a times 10 to the power of n, where a is a number between 1 and 10. For example, 0.00002 meters is written as 2.0 times 10 to the power of minus 6. Now, the order of magnitude. This tells us how many times bigger or smaller one thing is compared to another. Each order of magnitude is a factor of 10. So if a human cell is 50 micrometers and a bacterium is 5 micrometers, the human cell is 10 times bigger. That is 1 order of magnitude. So here is a worked example. We have a mitochondrion that is 5 micrometer and a ribosome which is 0.02 micrometer. So 5 divided by 0.02 equals to 250. The closest power of 10 is 10 squared, so that is approximately 2 order of magnitude. This type of question is very common in exams, and once you understand the idea, it's an easy mark. Now, let's look at the magnification formula, one of the most tested calculations in this topic. The formula is simply magnification equals image size divided by actual size. A simple way to remember this is the triangle. Image size on the top, magnification and actual size on the bottom. Cover what you want to find, and the formula show itself. Let's try an example. A cell appears 40 millimeters in a photo taken at 400 times magnification. Actual size equals 40 divided by 400, which is 0.1 millimeter or 100 micrometer. Another example, a nucleus is actually 6 micrometers but appears 3 millimeters wide. First, convert 3 millimeters equals 3000 micrometers. Then divide 3000 divided by 6 equals magnification of 500 times. The most important rule, always make sure both sides are in the same unit before you divide. That is the number one calculation mistake in this topic. Now let's look at the animal cells. Animal cells are eukaryotic, they have a nucleus. There are 5 structures you absolutely must know for animal cells: the nucleus, cytoplasm, cell membrane, mitochondria, and ribosomes. One thing to remember, animal cells do not have a cell wall, they do not have chloroplast, and they don't have a permanent vacuole. Each of these 5 structures has a specific function. The nucleus controls cell activities. The cytoplasm is where most reactions happen. The cell membrane controls what enters and exits. The mitochondria release energy through respiration, and the ribosomes build proteins. Let's now look at each one in more detail. Now let's look more closely at the nucleus. This is the largest organelle in the cell, and it is usually round or oval shaped. It is surrounded by a double membrane called nuclear envelope, and this membrane has tiny holes called nucleopores. These pores allow molecules like messenger RNA to move in and out. Inside the nucleus, the DNA is wrapped tightly around the protein called histones and form the chromosomes. In most human body cells, we have 46 chromosomes organized in 23 pairs. There is also a small dense region inside the nucleus that is called the nucleolus. This is where ribosomes are made. The nucleus is essentially the center, the control center of the cell. Now, the cytoplasm, this is a jelly-like fluid that fills the inside of the cell. This contains water, salts, sugars, and amino acids. And all these organelles are suspended within it. Chemical reactions, including anaerobic respiration, take place in the cytoplasm. The next is cell membrane. The cell membrane is a very thin layer made up of phospholipid bilayer. Bilayer means 2 layers. The phospholipid molecules have a hydrophilic head which loves water facing outward, and a hydrophobic tail which hates water facing inward. Proteins are small embedded in this membrane, acting as channels, carriers, or receptors. The membrane is selectively permeable, and this means it lets some things through and block the others. This is how the cell controls its internal environment. Now let's talk about the mitochondria. So these are the organelles that release energy for the cell. They carry out aerobic respiration using glucose and oxygen to produce ATP. They have folded inner membrane called cristae. These folds increase the surface area for reactions. Cells that need a lot of energy, like muscle cells, sperm cells, have many mitochondria. The more energy a cell needs, the more mitochondria it has. Now, ribosomes. The last organelle to know in animal cells is the ribosome. Ribosomes are the smallest organelles in the cell, so small that you cannot see them with a light microscope. They are found either floating freely in the cytoplasm or attached to a membrane called the rough endoplasmic reticulum. Their job is protein synthesis. They read the instructions from messenger RNA and join amino acids together in the correct order to make proteins. Every cell that makes proteins has thousands of ribosomes. And here is the key link. Cells that produce large amount of proteins will have very many ribosomes. For example, pancreatic cells makes a lot of digestive enzymes, so they have huge number of ribosomes. Always remember to link structure to function in your exam answers. Think about this. Let's pause here and think about something. You have a muscle cell or a pancreatic cell, both are in your body. Why does the muscle cell need more mitochondria? And why does the pancreatic cell need more ribosomes? Think about each cell does. The muscle cell it needs energy. Energy comes from aerobic respiration in mitochondria. So more mitochondria, more energy. And the pancreatic cell makes digestive enzymes, and these are proteins, and proteins are made by ribosomes. So more ribosomes, more proteins. This is exactly the kind of thinking the examiner wants to see. Always connect